You're still working, right? Good. Okay. So this is chapter 20, the lab material for chapter 20. So a lot of this stuff when we go over in the beginning, you should be somewhat familiar with, okay, when we talk about veins, arteries, capillaries, all right? So,
Part of those.
what, part of those?
We've heard of those.
Oh, yeah, you've heard about one or two of those? Well, arteries we know are going to take blood away from the heart, okay? But it's actually going to take blood from the heart, all right, to the tissues, okay? So, and in the tissues are going to be our capillaries here. Okay, so when we say it's going to take blood to the tissues, it's actually, when we're talking about the circulatory system, it's going to take it to the capillaries, all right, which live and reside in our tissues. So it goes heart to the capillaries, okay? Veins are going to take blood from the capillaries back to the heart, okay? So what are capillaries? They're an exchange. That's all they are. They're an exchange in which oxygenated blood is going to dump off its oxygen into the tissues, all right? Amongst other things, it'll bring nutrients like glucose and different ions and whatnot and dump it into the tissues. And at the same time, it's going to pick up waste. It's going to pick up carbon dioxide, all right? It's going to pick up stuff from the tissues, all right? So it's an exchange area, okay, between blood and tissues. And this is a huge, important adjective here. Porous. They're porous. Means that stuff can go in and out of them, okay? Like a colander, so to speak. Does anyone know what a colander is, right? Straining off ramen noodles. I got a cookbook for ramen noodles in case anybody's wondering. What? No, I don't. No. No. I was on a diet for a couple years in which I ate 2 peanut butter and jelly sandwiches every day for lunch. I had cereal, which was garbage, but it was sugared cereals because I was still eating Fruity Pebbles if I could, Fruit Loops, whatever. And then for dinner, I would do 3 or 4 scrambled eggs with cheese in it on top of ramen noodles. I did that for a couple of years. Yeah. Actually, you know, you're gonna laugh. I had I say this. I had horrible edema. I had pain edema, but just, there was so much sodium there. But I was in really good shape, you know, because I walked everywhere, ran. I was living down in the Caribbean at the time, and so it was. I honestly believe that if you walk, and they're somewhat active, not that you can eat anything, but you can sustain a relatively healthy lifestyle. I mean, I don't.
Sweat all day, every day.
I was. I loved it down there. Okay, so let's talk about blood vessels or even lymphatic vessels, but that's a different system. But vessels, but veins and arteries. All right, we're going to talk about the composition of these vessels, and so they're made up of layers. And again, I don't know what it is about nature, the rule of threes. There's 3, you know, 3 coverings to neurons, 3 coverings to the muscle. All right, it's always 3, 3, 3, 3. All right, so why are the vessels any different? They're not, okay? So we have 3 layers, and we call those layers tunics, okay? So of course it's easy to remember. You get the tunica intima, which is the internal portion, all right? Then the tunica media, you can probably guess, medium, middle, okay? And then tunica externa, the outside, okay? So I hope by now, okay, that you know this term here, okay? The lumen, that's the space. Here's our vessel, and then the space inside is the lumen, okay? If you don't know that, now you do, okay? That's where whatever fluid, blood, whatever we're going to have moving is going to be in the lumen space, okay? So the first tunic, all right, the innermost layer, all right, is going to be made up, there's that term again, endothelium. And we already know what endothelium is. Simple squamous epithelium. What does epithelium do? It sits on basement membrane. Okay? So it's going to be a simple squamous epithelium that'll sit on a basement membrane, but also here, when we're talking about the tunica intima, all right, it's going to have what we call a subendothelial layer, okay, which is areolar connective tissue. Areolar connective tissue is loose connective tissue proper. I won't write that out. Okay. So it's loose. A lot of ground substance, a lot of space for stuff. Okay. So this is the the innermost layer here. Okay. The middle layer, this is where you're going to find a lot of smooth muscle. Okay, a lot, and elastic fibers. Because if you're going to contract something, all right, remember, contraction is an active process. All right, relaxation is relatively passive. If you throw in some elastic fibers, now that relaxation, you can return the lumen to its original size. Okay, so if you get vasoconstriction and the blood vessel lumen gets really small, all right, as it's contracting, then it stops contracting and relaxes, it'll spring back to its original lumen size or diameter because of all these elastic fibers, adding to the passive process. Okay, so keep in mind, when we talk about vasoconstriction, we make the lumen smaller. If we talk about vasodilation, what do? Make the lumen bigger. Okay, all right, good. So contraction, vasoconstriction, relaxation, vasodilation. Know that relationship there, okay? Okay, the outermost layer is going to be the tunica externa. Again, we see a loose connective tissue, areolar connective tissue, loaded down with elastic and collagen fibers. The collagen fibers are nice. Collagen is the strongest of all the protein fibers. So it's going to help to support and offer somewhat of a protection. It gives it a little bit of a form there, okay? So we see a lot of collagen fibers here, but again, there's those elastic fibers again, okay? It's another protein fiber, and we all know what elastic does, all right? It'll help to give it that stretch and recoil, which is important, okay? So this layer, we'll see that it helps to anchor the blood vessel to, well, it could be to the bone, it could be to muscles, it could be to some other type of connective tissue. But we'll also see in this outer layer this structure right here, the vasovasorium. Some of these blood vessels are so big, they have to have their own blood supply or circulatory system. So that's what the vasovasorium is. All right, some of these large arteries, okay, mainly the veins, all right, will have their own blood supply. It's cool. It's funky, but it's cool. Okay? So let's take a quick look here, all right, at the anatomy of some of these. All right, so you can see we have our artery on our left and our vein on the right, okay? And one of the things that should be jumping out at you as you look here is, one, the thickness between, all right, the tunica media of the artery and the tunica media of the vein. The tunica media of the artery is much thicker, right? Because it has a lot more. This is smooth muscle, by the way. All right, also, something that should definitely jump out at you is this. Veins have valves. They have valves, okay? The valves prevent the backflow of blood, all right? It prevents it from pooling, and it helps it to move towards the heart. So we're not getting pooling in our legs, especially when we're younger, all right? Because when your muscles contract and relax, it acts as a pumping motion, and it pushes the blood up. It has to fight gravity somehow, all right? The valves help to prevent backflow. It'll push it up, the valves close, and it hangs there. And then the muscles contract again, your skeletal muscles, all right? When they contract again, it pushes it up, moves it closer to the heart, okay? So it's not the heart that's moving all the blood through your body, all right? The skeletal muscles help too. We'll talk about that later on, okay? So in between our two blood vessels here, between the vein and the artery, you've got our capillary bed. And if you look closely here at the capillary bed, you've got one layer of the endothelium, that's that simple squamous, and then you got a basement membrane. That's it. Thin. I can't even say paper thin, because paper is much thicker than a capillary. Okay, that's how thin this thing is. Okay, so we'll talk more about it, but also you can see a couple other things. All right, in between the tunica intima and the tunica media, you have this structure here called the internal elastic lamina there, or not lamina, yeah, lamina. All right, and that allows a little bit of elastic tissue stretch and recoil. Okay, and then we have another layer of it, all right, between the tunica media and the externa here. Okay, and then here's that vasovasorium there. You can see it much better over here in the wall of the veins here. You've got tons of them. Their own little blood supply. The artery has a little bit. All right, so Even blood vessels have their own blood supply. Okay. All right. So let's talk about a couple different types of blood vessels that you're going to see. All right. We're pretty familiar with the companion vessels, and just like the term describes, companion. All right. You have one and another. I'm trying to think of companions. You know, like a lioness and a lion. All right. So you'll have, when we talk about blood vessels that go out to your arm, radial artery. Well, you're also going to have a radial vein. That's what they're talking about. Okay. So you'll have arteries and veins that are going to supply the same part of the body. Okay, radial artery, radial vein, ulnar artery, ulnar vein, brachial artery, brachial vein, all that. Okay, femoral artery, femoral vein. Okay, so it's just going to be it's going to be an artery and a vein that go and supply the same area. Okay, so arteries, much thicker tunica media. All right, but they will have a much narrower lumen than the veins. Okay. But they have tons of elastic and collagen fibers. And that's why you can get veins collapsing on people. Okay? Has anyone here ever had to draw blood? Did you ever draw it too many times and collapse the vein? Did you? I didn't think you were going to admit to that. All right. Well, that's good because it's happened to me. On each other? Yeah. Right. It happens. So, but I mean, it happened. So veins will collapse because they don't have that high amount of collagen to help keep the lumen patent and the elastic fibers there to help to keep it open. Okay, so arteries do. So, but here's the part, and we'll talk about this if we have time today. All right, these vessels here, all right, can handle changes in blood pressure way better than veins can. Okay, way better. All right, because of the smooth muscle and because of collagen and the elastic fibers. Okay? Veins have a thicker tunica externa. Okay? They'll obviously have a larger lumen. Fortunately, they have fewer elastic collagen fibers here. That's why we get those collapsed vessels here. And then our last type here are the capillaries here. Okay? Here's the thing that you have to understand with the capillary. Remember we were talking about with the tunica intima there, you have that simple squamous epithelial layer, all right, the endothelium, basement membrane, and then the subendothelial layer, which is that loose connective tissue, the areolar connective tissue. Capillaries don't have that. Okay, they're the thinnest possible barrier you can have. One epithelial layer thick, and it sits on a basement membrane. Okay, and that's good because it facilitates the ease of exchange of respiratory gases, nutrients, and wastes. That's what you want. Okay. So that's what we're looking for. Rapid gas and nutrient exchange will occur there. Okay? So this diagram's in your book, and this basically is going to display and show us the different types of arteries and veins. All right? And we'll talk about them. All right? We got our elastic arteries, muscular artery, and then the arterial. Then on the other side, we have our venule, all right, small to medium size vein, and then a large vein. They're not really too clever with the naming of those. It's a vein, all right, small, large, medium. All right, and then we, of course, in between, we have our capillary bed. Okay, so let's start off talking about the arteries here. Okay, all right. So what we're going to see as we move further away from the heart, all right, obviously the arteries are going to become smaller, and they're going to branch more, all right, because we're going to start supplying blood to all these different regions throughout the body. Okay, so as we move further away, we're going to see more branching, and of course, as they branch, they get smaller. So if the artery gets smaller, then the lumen will decrease, and then we're going to start to sacrifice certain tissues or certain protein fibers. One of the ones that we start to sacrifice is the elastic fibers. Now you have to be careful with this last statement here. Increase in the relative amount of smooth muscle, meaning as we start to decrease in elastic fibers and other connective tissue that is made up of the blood vessel, okay, proportionally, our smooth muscle increases because if you're losing everything else proportionally, if smooth muscle stays the same, does it not by default increase? Kind of, sort of. Okay, well, it does. Just trust me. All right, but we will start to lose smooth muscle. I mean, large arteries like the elastic arteries and the muscular arteries will have a much thicker tunica media, all right, because of the smooth muscle, all right. But as it starts to get less and less, we get to the arterioles, all right, it starts to become thinner, but the majority of that vessel, more and more of that vessel will have more smooth muscle compared to the other tissues, okay? So that's why we say it increases in relative amount, okay? So our three types are going to be the elastic, all right, the muscular, and the arterioles, okay? So we start with the biggest one, the one that's closest to the heart, and that's going to be the elastic. So, example, the aorta, all right, is going to be an elastic artery, which means it's going to have tons of smooth muscle in it, but it's going to have a lot of elastic protein fibers, okay? So this, all right, or I should say these arteries, all right, are going to have tons of elastic fibers because, and here's the thing, when your heart beats, all right, and as you undergo systole and diastole, when the blood vessels or the arteries undergo systole, all right, they'll expand a little bit, okay, because you increase the blood pressure. But when it returns back to, when it recoils, as we go into diastole, you also kind of want to think of the vessel walls kind of snapping back, and that snapping back propels blood also, okay? So it's that passive kind of, it's a passive process because it's recoiling, all right? But it's also, we're getting an action out of it, okay? The actual active contraction of systole, all right, is pushing the blood. But then when the blood vessel walls, especially in the elastic arteries, kind of snaps back, all right, it gives it a little bit more of a push, okay? So it's kind of a cool system here, all right? So we'll also get some of that blood being propelled during diastole. All right, so here's some examples. The aorta, pulmonary trunk, common carotid, all right? Common iliac arteries, that's the arteries when the abdominal aorta splits off, down heading into the lower extremities, okay? All right, the next type of artery is going to be the muscular, also known as the distributing arteries. All right, now we're starting to branch off. All right, so we're getting smaller. We're going to take these arteries, they're going to go out to some of the different body regions here. Now this is huge because, all right, we call these the muscular arteries because there's a significant amount of smooth muscle in them, right? So this allows for vasoconstriction and by default vasodilation when we relax the muscle tissue. So these tissues will help with managing blood pressure changes, okay? So when we talk about blood pressure changes, they play a role, but, and we'll talk about it, not as much of a role as the arterioles do, okay? So here, all right, we were talking about these two elastic tissue layers, all right? The first one, the internal elastic lamina, is going to be in between the tunica intima and the tunica media, and then the external elastic lamina, and they're just like a sheet of elastic tissues, are going to be in between the tunica media and the tunica externa. All right, so these are the named arteries: the brachial artery, the radial artery, femoral artery, you name it, tibial, common, peroneal. All right, all those arteries that are named are going to be the muscular arteries here. All right, the last type here are going to be the arterioles. All right, now believe it or not, all right, these arterioles are like the last part of the arterial blood system before we get to the capillaries. So they're going to transition relatively quickly from a large version to a really small version soon. All right, so the larger arterioles will have three tunics. But when we get to the smaller arterioles, they look very similar to capillaries, all right, which means they'll have that endothelium there, but they'll have one single layer of smooth muscle tissue. And I'm going to show you a diagram here, all right, to help you out with this. But this is the part that's really important. You heard me talking about that vasomotor tone, all right? That's that sympathetic nervous system that's going to constantly be causing a slight amount of contraction, all right, on some of your blood vessels, all right, which will help to determine blood pressure, all right? So these smooth muscle cells are going to be constricted, not fully, all right, a little bit, and that's going to cause that blood vessel tone there that determines the blood pressure. So when we shut off the sympathetic stimulation to these cells, they'll relax and we'll get vasodilation, all right, which will help to decrease blood pressure. All right, this is all controlled, all right, in the medulla oblongata, okay? We'll talk about that later on. Okay, but what's really important is that these arterioles play a huge role in regulation of blood pressure and hence blood flow. Right? So when we talk about what's called the total peripheral resistance, when we talk about blood pressure, because blood pressure, all right, is equal to, all right, your heart rate times the total peripheral resistance. And the total peripheral resistance, this guy here, all right, is heavily influenced by that. Okay? Heavily influenced. Not all the way, right, but it's heavily influenced. Okay. So here's a quick picture of the histology of some of these tissues here. You can see here we're looking at an elastic artery. Look how thick, all right, the tunica media is. All this is smooth muscle, and it's going to be elastic fibers. You have your small single layer here of the tunica intima, but this is quite thick. Now we get into the muscular artery. All right, we still have, all right, our smooth muscle here, right, but it's not as thick. Then we get down to the arterioles. You can see very few, all right, smooth muscle cells, all right, that are surrounding the lumen here, or that are just deep to these endothelial layer. All right, capillaries. Capillaries, capillaries. All right, so blood comes from the heart, travels through the arteries towards the tissues. Okay, so when it gets to the tissues, it's in our capillaries here. Okay, and the capillary beds are found in between the arterioles and the venules. Okay, these capillaries, all right, remember, just think of them as leaky, I shouldn't say leaky, but porous tubes, like a straw. Okay, and that straw is just big enough to let a single red blood cell, all right, you can't have two side by side, only one can go down at a time. All right, now sometimes they'll get stacked up, and that's what this comes from. All right, that rouleau. Okay, but it's a single file. You can't have two together. Okay, which is good. Okay, we want to make sure that we can get, all right, because it slows the flow down slightly. There's a couple other factors that are involved in that, but it slows it down so oxygen can jump off, all right, and go into the tissues, and so that carbon dioxide can hop off. Okay. So here, these structures have that one single cell layer, that endothelial layer, the simple squamous, on top of the basement membrane. Okay? So again, those characteristics, the fact that the walls to this are thin, all right, and we have a small diameter of the lumen, is perfect for the exchange of respiratory gases, nutrients, wastes, and whatnot. Okay? It's very important. So we have three types: continuous, fenestrated, and sinusoid. Okay? And by looking at the names here, all right, we're talking about essentially how porous, all right, these capillaries are. Okay? This is the least leaky, this is the most leaky. Okay? Fenestrated is Goldilocks, it's right in the middle. All right? So here's our continuous, and if you look at it, okay, here's our simple squamous cells, and they're stuffed together, okay, and then we wrap them up in a nice basement membrane blanket here, okay? So you can see, all right, these lines here in between the cells, and we refer to those as the intracellular cleft, okay? They're not leak-proof, okay, but they're pretty gosh darn good about what they let cross over, all right, the capillary wall here, okay? So basically it prevents large molecules from escaping. Proteins are an example of that, okay? So if you're seeing a high content of proteins in your interstitial fluid, which is an example of edema, okay? If albumin, which is that protein we talked about when we were talking about the blood, all right, which is produced by the liver, and it plays a huge role in what we call the osmotic colloid pressure, how much of a pull stuff can get into the capillaries with. Well, if that is leaking out into the tissue, water is going to follow and you're going to get edema. Okay? These guys help to prevent that. Okay? So the erythrocyte, too big. Cells can't leave. They can't pass through these intracellular clefts. All right? So that's ideal. So the only thing that can pass through are going to be smaller molecules. Glucose is one. All right? Water, okay? Respiratory gases, but we know that respiratory gases can diffuse directly across plasma membranes. So that's not too much of an issue, though. Right? But they would rather go through a hole than have to diffuse through the phospholipid bilayer and then through another phospholipid bilayer. Okay? So basically, these continuous capillaries, all right, are only going to let really small things out. So we're going to see that in the muscle, skin, lungs. All right, lungs, big time. All right, for respiratory gas exchange. Okay? All right, the other capillary that we're going to talk about is the fenestrated. All right, when you have fenestrated, basically what we're saying is you have holes, okay, throughout the lining here, okay? And that's going to allow, in this situation, smaller proteins. In general, proteins are large, okay? But the smaller proteins will be able to leak across, right? Which is good when we're trying to deal with fluid transport, okay? Because certain fluids will follow those proteins, like I just said. When you have albumin leaking out of blood vessels into the tissues, water will follow it and other different fluids will follow that, okay? So we're going to see this type when we want to have absorption, right? Where do we want to have a lot of absorption? In the intestines, right? As we start to break down food and we want to absorb it from the lumen of the small intestine, large intestine, we want to pull it across and get it into our circulatory system. So we're going to see it here, and we're also going to see it in the kidneys. Because what do the kidneys do? What's a big function of kidneys? They filter. They filter the blood. Yeah, absolutely. They filter out the blood. Okay? And then eventually will form urine from the filtrate of the blood. Okay? The last one is called the sinusoid. And basically, I don't have a slide for that. But the sinusoid, you're going to see that like in bone marrow and the spleen. Right? That's when these capillaries have these like really big gashes in it. Okay? And a lot of times, if you're going to see it in lymphatic vessels like the spleen and whatnot, that's where we're going to actually allow the passage of some cells. All right? For example, hematopoiesis occurs in the bones. Well, we've got to get those cells that we just made into circulation. They can enter in through these sinusoid capillaries and then enter into the bloodstream that way. Okay? So they can start to circulate around. All right. What we're going to talk about now is this guy right here. This right here is a capillary bed. Okay, you can see in the background, here are some cells and whatnot. So we're going to talk about, all right, what a capillary bed actually does, what its function is. All right, so capillary bed is going to be found in between, all right, an arteriole and a venule. Okay, those are the smallest versions of arteries and veins. So that's where we're going to start to see, like, I hate to say mixing of blood, but that's where we're going to see, all right, a connection between the arterial system and the venous system. All right. So in our capillary bed, you've got one. So here's the vein, here's the artery, and then you've got like one kind of straight structure in the center of the capillary bed. That's the met-arteriole. Okay. It pretty much goes from the. If this is the, I'll put A here. This is the arteriole, and this is the vein here. All right. It pretty much goes from the artery to the vein. And then you'll have these little branches that come off, okay? Tons of little branches, which creates the bed here, okay? So on the end that's closest to the artery here, we're going to see smooth muscle cells, all right? All over it, along the diameter of it, you'll see smooth muscle cells all over the arteriole, okay? And then, all right, as we get further away towards the vein end here, or venule end, we won't see smooth muscles, okay? So if we look at the picture here, flip-flop what I just drew. Here's the arterial end. Here's the venule end. You can see smooth muscle cells wrapping around the arteriole. Here's the met-arteriole, all right, as it's going through. Okay, as you can see, all right, when we're closer to the arteriole end, we've got smooth muscles. here, and then along where these branches are coming off. That plays a role here in a second. Okay? You guys good so far? Okay, okay. You're just quiet. Oops. There we go. Okay, so the proximal part, all right, of the arteriole will have smooth muscle cells. As we get to the distal part, all right, we call that the thoroughfare channel. It takes on more of a vein type of characteristic, all right? So there's no smooth muscle cells there, okay? And then we're going to have all the branches, all right, that come off of that central structure, and we call that the postcapillary venules there, okay? All right, so let's check it out. Well, before I go, let me just go over this real quick. Here, let's show the picture here. Okay, so here's the met arteriole. All right, as we move through, then we call it the thoroughfare channel, and that makes that direct connection here to the venule. Okay, and then we've got our branches coming off here. All right, and we refer to those branches as the true capillaries. All right, and then the closer branches over here to the arterial side, you can see that there's smooth muscle cells wrapped around, all right, where these true capillaries connect, all right, onto the met arterial thoroughfare channel here, okay? And so if we constrict these smooth muscle cells, they'll restrict blood flow through these true capillaries. So it'll look like this, all right? So we contract them, and it squeezes and pinches off, all right, these true capillaries. So we're pretty much forcing the blood to flow through this channel here. Okay? When these smooth muscles are relaxed, then blood can flow all through these different channels or capillaries. You guys kind of see that? Okay? So those smooth muscles that we call those rings, we call those the precapillary sphincters there. Okay? So when they're relaxed, blood can flow through the true capillaries. When they contract, all right, they'll bypass the true capillaries and go directly through that thoroughfare channel. All right. All right. So we have this phenomenon known as vasomotion. Okay, and that's what we'll see. All right, where those smooth muscle sphincters will contract and relax, contract and relax. So they'll prevent blood from entering in the true capillaries for a moment, and then they'll relax, and then they'll allow blood to go through those true capillaries. All right. So basically, and this is an interesting number here, about 25% or a quarter of your body's capillary beds are open, which means, all right, what's this picture here? All right, 25% of the time, all right, or 25% of your capillary beds in your body look like this. At that same time, 75% of the capillary beds look like that. Okay? Which leads me to this term here. You definitely want to know it, perfusion. Okay? Perfusion, the term actually is how much blood enters into the capillaries, all right, and basically will wind up, all right, being exposed to the tissues. Okay, so we can have the exchange of nutrients and wastes. All right, so what we did was we quantified it. Okay, so it's the amount of blood entering the capillaries per, well, the volume per unit of time per gram of tissue. Okay, so you'll hear terms, and we'll talk about it about perfusion rate, right, when we talk about blood flow later on. Okay.
So the capillary bed is like an site of exchange?
It's going to be the site of exchange, yep, between our circulatory system and our tissues. And it goes both ways. Oh, it doesn't, yeah. Well, again, think of capillaries as just a site for exchange, right? Where the blood is going to dump off its nutrients, hopefully, into the, and other things into the tissue, and where the tissues will dump off their wastes and other things into the capillaries. Or if they're tissue that produces hormones, all right, they'll dump their hormones into those blood vessels, which can get it into our general circulation. So just keep it simple. It's a site of exchange. All right. All right, so now we're coming out of the capillary bed, and now we're going to head back towards the heart. So we have our venules, and those are going to be the smallest veins that we see, okay? And so similar to the arterioles, you'll have really small, thin venules, and then you'll have larger venules, and those larger venules will have the 3 tunics there, all right? They'll merge with the veins, okay, and then those veins will go from small to medium, then to large veins, and then back to the heart, okay? So our small to medium-sized veins, right, these are the ones that we see, the companion vessels, okay? So the vertebral vein, all right, will be the companion vessel for the vertebral artery, all right? Brachial vein, all right? The subclavian vein. Oh, no, not the subclavian vein, okay? All right, so as we get into the largest veins, they're going to be traveling with our elastic arteries. So an elastic artery would be the aorta. Well, an elastic vein would be the superior vena cava, inferior vena cava. Okay? So we'll see those. All right, but keep in mind, this is the big difference. Valves, people, valves. And its job is to prevent pooling. Okay? Again, we want to have one-way blood flow. You cannot have backflow, especially in the veins, because that will cause pooling. And when you get pooling for a long period of time, you increase your chances of clots. You increase your chance of clots, you increase your chance of CVAs, cerebral vascular accident, stroke, all right, damage to, you're going to get a pulmonary embolism, okay? That's bad, okay? All right, so the valves are going to be similar to the valves that we see in the heart, okay? They'll be made up of a decent amount of elastic tissue, but collagen, big time collagen, okay? One of my favorite facts about our circulatory system is on this slide here, okay? That when you're sitting here, like we all are, I'm the same way, that 70% of our blood is sitting in our circulation. But a significant amount of that blood is sitting here in the veins. That's crazy, all right? That is nuts and crazy. That's half of the blood volume in your body. So on average, 5 liters, all right? That's 2.5 liters of the blood that's sitting in your body is going to be sitting in your systemic veins, all right? Only 12% is in the heart, okay? 18% is going to be in your pulmonary circulation, going to the lungs. Not a lot. All right, look how little are in your systemic capillaries, 5%. It's this number here, that 55%, all right, is going to be in the veins. So, me saying that, I want you to consider the veins as reservoirs for your blood, okay? Reservoirs for your blood. So if we want to decrease the reservoirs, we want to push the blood out of the veins, maybe into the tissues somewhere. All right, and if we want to increase, all right, the amount of blood in your veins, then we're going to want to maybe pull the blood from the tissues or other areas of your circulation here. Okay, and we can do that. Okay, so when you see on this slide here the term vein, think of reservoirs. Okay, so if we want to move blood from the veins out of these reservoirs, we just have to vasoconstrict those veins. Okay? It's harder to jam blood into vasoconstricted veins. So it'll most likely stay out into the tissues or some of the other circulatory structures. Okay? If we want to get the blood back into our reservoirs, simple. Open up the pipes. Vasodilate.
So you need like a stimulant.
Some sort, yeah, depending on what it is. Well, think about it. Like when you're exercising, all right? We can't have blood just hanging out in your veins. Your muscles need it. So the veins will constrict, they'll vasoconstrict, all right? And that will force, all right, blood into the circulatory system, all right, mainly into the arterial circulatory system, then out to the skeletal muscle when you're exercising. So we'll see that vasoconstriction there, okay? But like all of us, we're sitting here. We're doing this. Our veins are vasodilated. So it's sitting there. That's why when you take a flight somewhere, especially a long flight. If you ever fly to Australia, you know, one of those 17-hour long flights, what do they tell you? Get up and walk around, because your blood is pooling there in your vascular system, in the venous system specifically. All right. So when you get up and walk around, what's going on? Your skeletal muscles are contracting and relaxing, pumping the blood from the veins, all right, and moving it through with the valvular system in the veins and getting it out of that reservoir. Okay. All right. So when we talk about the pathways to blood vessels, there's two types. There's the simple pathway, and then there's the alternative pathway. So this picture here, all right, here's the first type. You can't see, hold on. I know you can't see. Okay, simple pathway is pretty much what we've come to learn so far. Okay, you got an artery that goes into a capillary bed, and then a venule leaves and goes back to the heart. That's the simple pathway. Okay, we have a couple of alternative pathways. We'll go through this. All right, you may have heard the term anastomosis, and that's where you'll get connections between different blood vessels. They'll attach to one another or have pathways or alternative pathways between blood vessels. The first one is called the arterial anastomosis. The second is the venous anastomosis. Then you can have an arterial venous anastomosis, which is almost like a shunt. It'll completely bypass the capillary bed. And then there's the portal system. And we saw that in chapter 17 when we were talking about the hypophyseal portal system between the hypothalamus and the pituitary. Remember the adrenopituitary? All right, the hypothalamus would have regulatory hormones, and most of those would go to the anterior pituitary through the blood system. That was a portal system. All right, we had two capillary beds. One capillary bed in the hypothalamus, the other capillary bed in the pituitary. So you were already learning this and you didn't even know it. Okay? So where am I? Simple pathway. Okay? So real simple, okay? You have one major artery that's going to deliver blood to a region or an organ, and then you're going to have one major vein or one vein take it away and return it to the heart. Perfect example that we talk about is the splenic artery, okay, which we'll see when we start labeling. Splenic artery is a branch off of the celiac trunk, goes directly to the spleen, goes into the organ, and then you have the splenic vein that leaves and goes into the back to the venous system there. Okay. So we essentially call these types of arteries an end artery, which means it ends at the organ. Okay, you'll stop there. Okay. All right. So we already talked about the simple pathway. I feel ad nauseum. All right, we have our arterial that goes to the capillary bed, and the capillary bed drains into the venule. And the venule is going to move into one of the major veins. Okay, pretty good about that. You feel pretty good about the simple pathway? I like it too. I wish it was that easy. And now we're going to go into the alternative pathways. All right, but again, keep it real easy. All right, arterial anastomosis is where you're just going to have a bunch of arteries joining one another before, all right, we get to the capillary beds. So we see this, all right, the example that we use here is going to be the superior and inferior epigastric arteries, which are going to supply the abdominal wall. We'll see that with the mesenteric arteries. All right, a lot of arterial anastomoses we'll see in the abdomen, the digestive system. Okay, tons and tons. All right, so venous anastomosis. All right, these guys, again, you're going to have one artery leading into the capillary bed, and then you'll have a bunch of veins that are going to join up with one another. And we see this specifically when we get into the blood vessels of our arms and legs. You've got the basilic vein, the cephalic vein, the brachial vein. All of those veins drain your upper extremity, okay? And they'll eventually dump all of their blood into the axial and subclavian veins, okay? So we'll see very similar when we're down into the lower extremity, all right? So again, that's why the venous anastomosis is much more common, okay? An atrial venous shunt, all right, basically we see this, and I love this structure in the fingers and toes, all right? And you'll have these arches here, okay? One will be a venous arch and one will be an arterial arch, and it sends out these little tributaries into your digits, into your fingers, into your toes. All right, and you'll get a mixing of the blood, especially when you get the return blood that comes back. All right, but these are great because we can shunt and cut these types of systems off, especially when we enter into like a hypothermic type of situation. For example, if you fall out of a boat and you're in the middle of a frigid cold lake, like Lake Ontario or Lake Placid up in New York, what happens? All right, your blood starts to get shunted away from your fingers and toes, and it's going to go towards your actual internal organs to keep those organs warm and give them the nutrients that they need. At the same time, that's the early on. I mean, you probably have been experiencing here if you stood outside recently for longer than 10 or 15 minutes. Your fingers get cold, your toes get cold, because your body is starting to shunt away, all right, the blood from those areas because you're starting to get hypothermic. All right, and then the portal system, we talked about that. You got 2 capillary beds. All right, so we start off with the artery, then we get a capillary bed, then we get to the portal vein, and that's when we start to see a little bit of mixing of blood. All right, then to the capillary bed, then back to the vein. Okay, we'll see this also in the liver. Okay, you have the hepatic portal system, and we'll talk about that a little bit. But we already saw it when we were talking about the endocrine system with the thalamus and the pituitary gland there. Okay, the hypothalamo-hypophyseal portal system there. Sorry, I'm just thinking here. All right, let me do this one here, and then we'll call it a night, I think. All right, pulmonary circulation. Quick review. We've already talked about this, okay? So let me walk you through it real fast. The right ventricle has deoxygenated blood that it received from the right atrium. The right atrium pretty much gets all the blood from your body. Okay, superior vena cava, inferior vena cava, and the circulation or the blood from the heart, the coronary. Remember, the coronary sinus will dump its blood into the right atrium. So all that blood heads towards the lungs. Okay, it's deoxygenated. Okay, exits the right ventricle through the pulmonary trunk. Okay, and then from the pulmonary trunk, all right, it splits into left and right pulmonary arteries. Okay, which means they'll go to the left and right lungs respectively, all right? So it goes to the lungs, and these arteries will start to divide into smaller arteries, eventually arterioles, and then eventually into our pulmonary capillaries that surround the alveoli. And that's where we'll get our gas exchange. So that it's deoxygenated blood, so carbon dioxide will leave, oxygen will come into the blood there. At that point, now we have our oxygenated blood, okay? So then the blood will then leave, all right, the capillary beds and the alveoli, all right? And then it will head towards the venules, then to the pulmonary veins, and then back to the left atrium, and eventually into the left ventricle, then into the aorta, okay? That's our pulmonary circulation. If you are not quite sure, all right, about pulmonary circulation, look at this slide, okay? It's basically putting into words what I've talked about before, okay, in class. Let me just do, let me do this one right here, and then I promise you, this is the last one, all right? Coronary circulation here, all right, basically, all right, as blood moves through the left atrium into the left ventricle, it's going to get pumped out into the atria, all right, through the semilunar valves. Immediately on the other side of the semilunar valves in the ascending aorta there, all right, we have the openings for the left and right coronary arteries. Okay, so that's the only branch off of the ascending aorta are going to be the left and right coronary arteries. And as you know, those arteries supply the heart wall. Okay, and then eventually the ascending aorta will turn into the aortic arch, in which we get these 3 vessels that come off of it, but we'll talk about that on Monday. All righty, any questions for me? We did a lot today, you guys.